I am pleased to introduce our first speaker for today, and that is Dr. Rodney Johnson. Dr. Johnson is a professor of integrative immunology and behavior, and he is the head of the University of Illinois Department of Animal Sciences. And he was actually a recipient of the ILSI North America Future Leader Award some time ago. So Dr. Johnson will kick off the series with an overview of the information we'll hear over the 5 webinars this month. So Dr. Johnson, we'll turn the platform over to you.
Okay, thank you. Can you see my screen?
Yep, looks clear.
Okay, great. Well, first, let me just thank everybody for taking time to join us today, and I hope you'll mark your calendar and join us each Friday for the next several weeks. And you're going to hear from a number of faculty at the University of Illinois that participate in the Division of Nutritional Sciences and the work that they're doing. And you're going to hear from a number of our students, and which I think is a tremendous opportunity for the students to kind of get out there and participate in this type of format and talk about their research, regardless of the stage that they're at in their graduate programs. But I want to first give a shout out to Dr. Elvira De Mejia. Dr. De Mejia is the current director of the Division of Nutritional Sciences and organized this webinar series that is brought to you in collaboration with ILSI North America and the American Society for Nutrition. Okay, I'm just going to provide a very brief overview, and that's, I was asked to kind of introduce this webinar series, and I'm happy to do that. Obviously, everybody recognized today that there's bidirectional communication that occurs between the gut and the brain. And this has really been one of the most interesting and dynamic fields of investigation that I've seen, having been on faculty here for almost 30 years, just the way this field evolved so rapidly. And I was a bit of an outsider because I'm not a microbiologist, and in a moment I'll show you, I kind of actively resisted getting into this field, but it was so compelling, and it attracted me and others to get involved to some level. Shown in the graph is the number of papers that are revealed with a simple PubMed search using gut-brain axis as keywords. And you can see that in 2019, the last year of where we have complete data, there was almost 475 papers published that were connected to the gut-brain axis. And if you go back one decade to 2009, only 16 papers were revealed. And so just tremendous exponential growth and interest in this area. And I think, the driving factors are shown here, in my opinion, the emergence of next generation sequencing technology to characterize the gut microbiome, studies, showing behavioral phenotype can be transferred via gut microbiota. And this was really just stunning to me. You'll remember the studies where taking microbiota from an obese animal and putting it in a skinny animal and creating that phenotype. Really amazing results. Probably wondering what the mechanisms were at that point in time, but nonetheless, it really was attractive and created a lot of interest. And then, of course, studies showing the correlation between the gut microbiota and neuropsychiatric disease, and then just the sheer recognition of the potential to manipulate the gut microbiota in order to improve health. So we know that the microbiota is highly pliable, and I think you're going to see that through the course of this webinar series, including today, where Dr. Woods will talk about how the exercise can affect this communication system. So here's one of the reasons I suppose I was actively avoiding, the microbiome field. I was busy, doing my own work that I had been working on for many, many years. And we were interested in how the peripheral immune system talks to the brain to induce the behavior that's illustrated in that oil painting entitled The Sick Girl. And you can see the classic behavioral signs of an infection that all of us have experienced at some time or another when we develop some type of infection. So if you start at the bottom, this is just putting it into the classic inflammatory pathway where you have inducers. So bacteria, viruses have certain molecular patterns. that will be recognized by sensory cells. And we're talking about the immune system, and the easiest one to appreciate, of course, is the macrophage with the expression of toll-like receptor 4. When that receptor is activated, those sensory cells are going to produce inflammatory mediators, things like pro-inflammatory cytokines, prostaglandins, histamine, and then these inflammatory molecules can access the brain either through a humoral pathway where they enter circulation. They can be actively transported across the blood-brain barrier, or they can go to brain areas that are devoid an intact blood-brain barrier and actually passively diffuse into the brain parenchyma and interact with different cells of the brain. The neural pathway involves the vagus nerve, and we now know that the vagus nerve expresses receptors for certain pro-inflammatory cytokines, and there's a neural signal then that can connect the peripheral immune system to the brain. Now, what's important from our perspective anyway is that whether it's a humoral pathway or a neural pathway, they both have in common the stimulation of microglial cells, which are the macrophage-like cell that resides in the brain. And notice that they're a significant proportion of the cells in the brain, so accounting for about 15%. And it's these cells that when they become activated, start producing pro-inflammatory cytokines that elicits the sickness behavior. So in my lab, we were interested in microglial cell activation and its role in inducing sickness behavior. We also have a lot of interest in microglial cell activation and how it can affect brain development. We study this both in fetal brain and in the early postnatal brain, and then how aging and microglial cell activation is manipulated, or how aging affects microglial cell activation. And what I'm simply showing in this graph on the right is if we isolate microglial cells from an old mouse compared to a young adult mouse, what we see is a phenotypic shift in the microglial cells so that they are more pro-inflammatory. So old but otherwise healthy animals have kind of a low-grade inflammatory reaction in the brain that we think contributes to things like cognitive aging, maybe a predisposing factor to neurodegenerative diseases, things of that nature. Incidentally, we've recently shown that this can be somewhat corrected through the use of dietary fiber. So this is where I started to become interested in, the microbiome, because all of a sudden this fantastic work that was occurring in an area somewhat different than my own kind of swerved into what we were doing. And this was an important paper for our lab because it demonstrated that the host microbiota can actually control the maturation and function of the microglial cell. Again, we didn't know much about the mechanism of how this is occurring, but clearly you can see down in that lower box some of the findings that came from this paper, where germ-free mice had immature microglia. Their microglia tended to be ramified and inactivated, and that's what's shown in the reconstructed microglia there on the right. You can see the ramified morphology of when it's in the germ-free mice. There was a reduced or increased density of microglial cells, but they didn't work right either, so they had a dampened innate immune response. And all of this could be corrected by reconstituting the gut microbiota. So clearly indicating that the gut microbiota have the ability to affect this very important immune cell that resides in the brain. And so you can imagine then the interest in understanding the pathways that can control this communication. So here, the question is, how does the gut microbiota talk to the brain? How do we go from understanding that these changes in the population of microbes in the gut are actually causing fundamental changes in behavior? So of course we've had some appreciation for this now, and that the gut microbiota produce bioactive short chain fatty acids like acetate, butyrate, and propionate. These short chain fatty acids can be absorbed and therefore enter circulation. However, they're fairly short-lived, detected at relatively low concentrations, and there's really not a lot of evidence to suggest that at physiological relevant levels that they actually enter the brain, and moreover, the receptors for the short chain fatty acids not expressed in the brain. but they have this other mechanism where they can bind G protein coupled receptors like FFAR2 or 3. And importantly, cells of the immune system express these receptors, and the vagus afferent nerves do as well. So now if we think back to my last slide where I was talking about the immune system communicating with the brain, where it involved both humoral and neural pathways, we have the same possibility in this gut-brain axis, where in the humoral pathway, it's probably not, the short chain fatty acids directly accessing the brain, but rather inducing hormonal changes in the periphery, regulating immune function, and that those molecules then have the potential to change the way the brain is functioning. And then we have the neural pathway, this viscerosensory signaling pathway. And this is important because the vagal afferent nerves project to the nucleus tract solitaire, shown here as the NTS. And in this area, neurons project to the limbic system, including areas such as the amygdala, which is involved in regulation of emotional behavior. So the things like anxiety and fear, pain, and social behavior, all of which now have been shown to be affected by the gut microbiota. So the pathways of communication are starting to be illuminated, which creates even better opportunities for manipulating this pathway to improve health. So this is it's a complicated story, right? Because we're talking about the brain and neural and humoral pathways and changes in the gut microbiota. And so you can imagine it takes a team, if you will, to kind of train and prepare future leaders, to address the complex issues like this. And so when we at Illinois set out to develop a training program, what we did is identified faculty who had an interest in the gut-brain axis or working at different levels of this issue, and we divided them into areas of expertise. So we have 12 preceptors, 12 faculty shown there on the far right, divided into 3 areas of expertise depending on what they were doing. Some focused on the brain, others on the gastrointestinal system, and others on the gut microbiome. With this training grant that was funded through USDA, this National Needs Graduate Fellowship grant that's titled here, Nutrition and the Gut-Brain Axis, Implications for Development of Healthy Aging, we're supporting 8 fellows. 3 are funded through the grant itself, and 5 others are being funded through different units here on our campus. So it's really a tremendous opportunity to bring lots of people together with a common interest. All of the students in this program have a co-advising model where they have to have at least 2 faculty mentors representing 2 of the 3 different expertise areas. So really building in guarantees for interdisciplinary training. We've developed coursework to support the program, areas that relate to professional development, and importantly, they require a experiential learning event outside of the university. So finally, just to kind of show you where we're going the next several weeks, next week, Dr. Kelly Swanson and his student Celeste Alexander will talk about sex-related differences in host metabolism and health. The following week, Yanina Pepino and Clara Salme will talk about their work on low-calorie sweeteners, treat or trick. The following week, Aditi Das and Andrew Steelman will talk about their work relating to a neuromodulatory role of omega-3 fatty acid endocannabinoids found in the gut. And then September 25th, Dr. Sharon Donovan and Naiman Khan will talk about their Strong Kids 2 program, specifically the role of childhood nutrition and obesity in the microbiome brain axis. And today, we're going to hear from Dr. Jeff Woods and his student Noah Hutchinson on the effects of exercise, diet, and the gut-brain axis. So with that, again, I want to thank everybody for joining us, and I'm going to turn it back over to Maria to introduce Dr. Woods.
So moving on to Dr. Jeff Woods. Dr. Woods is a professor of kinesiology and community health with additional appointments in the Division of Nutritional Sciences and the new Carle, Illinois College of Medicine at the University of Illinois. So Dr. Woods will be sharing information about his work on the influence of exercise and diet on the gut-brain axis. And just a reminder to go ahead and type your questions at any time. So Dr. Woods, we'll turn the platform over to you.
All right, thank you so much, Marie. I definitely want to thank everybody for attending here on this Friday afternoon. I also want to acknowledge the Division of Nutritional Sciences, as well as ILSI, for the opportunity to share our presentation with you today, and especially for our students, in particular today, Noah Hutchinson, my student in nutritional sciences, who really appreciates the opportunity to present, especially during this pandemic when we don't have too many opportunities to get out there in front of people. So for students listening in, shameless plug here, we're always looking for bright, motivated students to come to Illinois and learn and work with us. If you're interested, please make sure that you check out our website shown here on the slide, and email any of the faculty in the Division of Nutritional Sciences and express your interest. And we've got a great group and we'd love to have you. So today, really, I'm going to talk about exercise, some about diet, but certainly exercise. I'm a kinesiologist by training. I am not a microbiologist like Rod, and I came into this field about 10 years ago with interests because of my interest in inflammation and the immune system. And really, you can see on this slide many different paths of communication between the gut, including its microbiota, and the brain. Some we know about, as shown here. Many we don't. Anybody who's ever had a stomach ache knows that the gut can talk to us. But I think it's really important to remember that this dialogue is two-way. So it's not just the microbes and their products or the immune system in the gut talking to the brain. It's also the brain talking back to the immune system and also to the gut. And so there's this bi-directional communication that goes back and forth, regulatory feedback that leads to alterations in health of the gut, the brain, and the whole body for that matter. For example, gut metabolites can cross the barrier or be recognized by receptors on the barrier to generate signals in the host. Butyrate can bind to free fatty acid receptors 2 and 3 to induce GLP, which acts on the brain to induce satiety. So that's just an example. In the opposite direction, hormones produced by the host can affect the gut microbiota and gut health. Noradrenaline released by adrenergic nerves can interact with certain gram-negative bacteria in your gut and cause them to grow or be more virulent. Indeed, chronic stress, which increases norepinephrine, can cause dysbiosis and gut disease. So I think we all know that diet certainly has a big effect on our gut microbiota and its function. What you may not realize is that exercise does as well, and so that's where I'm going to focus today. Diet and exercise affect who's there, in other words, which microbes are in the community, and what they do, what products they produce, and how they interact with the host and each other. And we know that diet and exercise influences brain structure and function. So could it be that some of the effects of diet and exercise are mediated through the gut microbiota and its metabolome? So we do know that exercise affects the brain. I've been involved in some research here at Illinois for a long time with a guy named Art Kramer. He's moved on, but we published some articles back about 10 years ago now linking exercise training to changes in brain structure and function. You can see the titles here. Exercise training increases the size of the hippocampus and improves memory. Some of these effects are mediated through BDNF, or brain-derived neurotrophic factor. In people, we can measure that in the blood and correlate it to changes. Of course, those are correlational, and we would need to do more mechanistic studies to rule that in as the mechanistic pathway. So really today, what I want to do, for those of you not familiar with the gut microbiota, I'll give you a quick catch up on that. And then I'm going to talk about a study where we address the question, does exercise independently affect the gut microbiome? And independently, I mean independently from diet. I'll share with you results from really the first human longitudinal trial on exercise in the gut microbiome. Thirdly, I'm going to focus on a study where we addressed the question, does exercise-induced changes in the gut microbiome affect host health? And this is a study we did in collaboration with Mayo Clinic, where we did the first exercise biota transplant into germ-free mice. So we had mice that were sedentary, mice that had exercised, and then we transplanted biotas from those animals into germ-free mice that did not exercise, and then hoped that some of the effects of exercise would show up in these animals just by transplanting the biota. And then lastly, Noah is going to talk about some of our current work and our future directions. So I'll talk about stuff we've done, and Noah is going to talk about stuff we're doing and are aspirationally thinking about. We've got a gut barrier. Gut barrier consists of epithelial cells, colonocytes, shown here in blue, that act as a barrier between the outside world, which is the lumen of the gut, and the inside world, which is us. In the lumen, there is fecal material, there's luminal bacteria. We've also got a barrier of mucus, this mucus layer that exists kind of right above the epithelial barrier, and that's very protective in terms of allowing protection of the barrier. But it's also important because there's mucosal associated bacteria that sit on the mucus and use it for food, and also interact through the barrier there. There are other cells in this barrier, including goblet cells, which secrete mucus, Paneth cells, which secrete antimicrobial peptides, enteroendocrine cells that secrete hormones, M cells or microfold cells that actually sample the outside world and mucosal environment and lumen to educate our immune systems against our microbiota, to train it so that we don't react against everything in the microbiota. The barrier is characterized by tight junction proteins that exist between the cells. So this is a very tight barrier. Things do get across, either actively transported, of course, or leak through via diffusion. The barrier can be broken in cases of, for instance, inflammatory bowel disease. And when that happens, some of the outside world, some of these bacteria and the products can enter into the lamina propria, the area just below the barrier. And right below this barrier, you can see there's a lot of immune cells, which you obviously predict to be there, because this is kind of at the interface between the inside and outside world. Cells like macrophages, neutrophils, and dendritic cells that, if the barrier is broken, can have a profound inflammatory response that can cause further barrier breakdown, but also systemic effects, including sickness, behavior, and changes in the brain. So the gut microbiota, or our flora, consists of bacteria, archaea, fungi, and bacteriophages. These bugs can be commensal, mutualistic, or opportunistic. There is a very competitive environment in our gut. There are niches of bacteria in very different spaces along the longitude of our gut, but also along the cross-section of our gut and the mucus layer, for example, and within the lumen. And they fight each other. They compete for resources. So it's very important to kind of recognize that we have this ecosystem living in us that is in constant flux and competing in and of itself. So they're competing for resources within that environment. Each person's biota is unique. For example, in the human colon, there's about 500 different species of bacteria. I will say that a person's biota, while unique, is relatively stable in adult life in terms of the relative abundance of different bacteria and types and species present. These things can change, and I'll talk about that in a second. You may have heard the term microbiome. So microbiome, it differs from microbiota in that it really is the genetic makeup of the biota. So looking at the biota through its genes, we call that the gut microbiome as opposed to the gut microbiota. Also remember, we have a skin, oral, and vaginal biotas too. So today we're really just focused on the gut biota and the gut-brain axis. So what do gut microbes do? Within the gut, they compete for resources and survive, like I mentioned. They produce metabolites, enzymes, and hormones. We've already talked about short-chain fatty acids as being one metabolite, important metabolite. I'll hit on this later. They detoxify some toxins, and they affect the gut barrier, either good or bad, depending on the taxa that we're talking about. They communicate to our body, and so we do know that gut bacteria and their products can promote maturation and development of our immune system. They can promote or inhibit inflammation. They can certainly regulate metabolism and energy harvest. They have a direct link to the liver through the hepatic portal circulation, and they communicate through the gut-brain axis. So more and more, we're beginning to realize the importance of the gut microbiota in overall human health. So an example of this, and I think Rod mentioned this, is a study that was done in science by Verdura and colleagues in Science in 2013, where they actually took feces from twins, either an obese twin or a lean twin, and they did a fecal microbial transplant into germ-free mice. And lo and behold, the animals that received the fecal microbiota from the obese twin had a change in fat mass over the course of 15 days, such that there was a significant difference between those that received the obese fecal microbial transplant and those that received the lean microbial transplant. So that's really powerful evidence, the power of poop, that changes in the gut microbiota can lead to phenotypic changes in the host. And this was one of the first studies to show that surprised everybody. But then there's been many studies since, including some with respect to the brain and behavior. Currently, the only FDA-approved use for fecal microbial transplant is for a disease called Clostridium difficile, where it shows a greater than 90% cure rate. So for other applications like the treatment of obesity or Parkinson's or other things, investigational new drug status is needed to be able to do that, because there are some risks associated with taking someone else's feces and inoculating it into you. To understand the gut microbiota, we need to answer a few questions. We need to know who's there, and so we can use next generation sequencing in order to do that. We need to know what they're producing, so we can fall on metabolomics or targeted metabolomics to be able to address that question. And then, of course, we need to know about the mechanisms of communication. So this could be diffusion, either way, again, remember, bidirectional, receptor mediated. Examples we gave were of free fatty acid receptors. Could be neurally mediated, or it could be epigenetic. So we need to understand a lot of these things in order to truly say that a certain aspect of the gut-brain axis is operating and controlling human health. So I think we're all pretty much aware now that the gut microbiome is pliable. Many things can affect it. Bacterial exposures at birth and early life, your genetics plays a role. Antibiotics certainly can disrupt your normal flora. The environment that you live in, whether you live with pets, where you live, your hygiene, gender makes a difference, age. Usually it takes about 1 or 2 years to firmly establish your unique microbiota. It's fairly stable throughout life. It's kind of like your unique signature, which from a research standpoint is important, because it's really hard to compare across individuals. It's hard to compare me to you because we have different biotas. And so it's very important to do repeated measure studies to be able to compare, what happens to a microbiota when you change lifestyle, for instance, when you exercise or when you change your diet, because it's different between people. And so with that, I think, we all know that diet, including changes in dietary fiber and macronutrients, can have a big impact on your microbiota and its products. But what about exercise? I think that's kind of one of the unique things about my lab in the last 5 or 8 years that we've contributed to the literature. We've clearly shown that exercise can influence the microbiota, and I will show you that data. So some of the questions that we've been addressing really surround the role of exercise. Is exercise medicine for your gut-brain axis? So the questions in green, I think I'll address today, and I'll show you some data that exercise does affect the gut microbiome and metabolome, and it also does so independent of diet. Questions we're working on, and I have some data in the colon today, colon health. Are exercise-induced changes in the gut microbiome and metabolome responsible for the beneficial effects of exercise on the brain and other tissues? And then the ones down at the bottom in red are studies we want to do in the future. For instance, can exercise synergize with diet to beneficially impact the gut microbiota? And if so, what types of exercise are most important? Are they different? And does it do so also for those with microbial dysbiosis? So Noah's going to talk briefly about a study that we're just starting looking at the effects of exercise in IBD patients. So what do we know about exercise in the gut? Well, if you look here at incidence rate relative to dose of physical activity increasing from left to right, you can see that gallbladder disease, colon cancer, constipation are all reduced by increasing levels of physical activity. There is a point of no return, however, for gastrointestinal symptoms. If some people cross over a certain level of physical exertion, highly intense, prolonged exercise, for example, in the heat, they can actually realize gut symptoms, and that may be due to some changes in the gut barrier as well as other changes. But regular moderate physical activity tends to help the gut in terms of certainly reduction from colon cancer, for example. One thing you don't see on this list is inflammatory bowel diseases like Crohn's disease and colitis. And this study was, this review was published back in 2001. And really not much has changed in the last 20 years, unfortunately. That's why we want to do that study with exercise and IBD. But there is reason to believe, at least based on some of our data in healthy folks, that exercise could be beneficial to people with inflammatory bowel diseases. So we got into this field. I think Rod told a little bit about why his lab got interested in that. And this is the paper that got our lab interested in it. So I had a student, Mark Cook, back in about 2010 or 11, try to answer the question whether exercise is beneficial for experimental colitis in mice. So this is a model of dextran sulfate sodium, DSS. We give that in the drinking water and the animals get colitis that mimics human colitis, although it's still a mouse model. And he found, and you can read the title of this paper, he found interestingly, paradoxically, that forced treadmill exercise exacerbated inflammation and caused mortality in this model, while voluntary wheel running actually was protective. So there was a weird dichotomy in terms of exercise. And it's weird because, you know, many people use forced treadmill exercise training in animals, rodents, mice, rats, to improve the health of many tissues. But in this model, we found that it actually was not beneficial. But if you give animals access to a voluntary running wheel, it actually is protective in this mouse model. And so that led us to study the microbiome, because we do know that the microbiome and microbiota is related to colitis and Crohn's. And so this was kind of our rationale for getting into this field. So what do we know about how exercise affects the gut microbiome in animals? And you can see the titles of many different studies. This was a couple years old now, and a couple done by us. And so as a direct result of that colitis study, my student Jacob Allen, who is now a professor here at the University of Illinois, assistant professor, did show that voluntary and forced exercise differentially altered the gut microbiomes in the same strain of animals where we saw the differential effects on colitis. We also did some studies with a colleague at Mayo Clinic examining the effects of diet and exercise, and found that it orthogonally affected the gut microbiome and was associated with differences in anxiety and cognition within the gut-brain axis. So understanding the overall community structure is important in studies of this type, but it's really the products of those communities and their interactions with the host that will be important in explaining gut microbiota effects on the host. So one of the first studies to kind of look at this was done back in 2008. And what they found was that in mice that were given running wheels, they showed that it altered the microbiota composition, but it also increased butyrate concentrations in their cecums. And you can kind of see that here, about a twofold increase in butyrate in the wheel runners versus the sedentary controls. And they also looked at, using kind of old technology, they looked at the microbiome, and they showed that there was a couple butyrate producing strains that were elevated in the wheel runners when compared to the sedentary controls, SM7/11 and T2-87. So this is really the first evidence then that exercise might have an independent effect on short chain fatty acid production, at least in rodents. So why are short chain fatty acids important? Well, first of all, short chain fatty acids include acetate, propionate, and butyrate, 2, 3, and 4 carbon molecules. They're primarily produced as a result of the digestion and fermentation of dietary fiber by certain gut microbes. And these short-chain fatty acids then can communicate through the host through free fatty acid receptor 2, 3. They also act through histone deacetylase inhibition to cause many different host effects. For instance, we harvest about 10% of our energy in the colon in the form of short-chain fatty acids. These short-chain fatty acids also can affect gluconeogenesis and lipogenesis. They can increase insulin sensitivity. They are food for our colonocytes, so they maintain a healthy barrier, butyrate being especially important in this regard. They can regulate satiety peptides like GLP-1. They can regulate our immune system. They can stimulate mucus secretion. And as Rod pointed out, they can also alter your brain in terms of impacting microglia and memory. Couple enzymes that are important for the biosynthesis of short-chain fatty acids include B-coat, butyryl coenzyme A acetate CoA transferase, so B-coat for short, and methylmalonyl CoA decarboxylase, or MMDA for short. So in the data that I'll show you, in addition to the gut microbiome analysis that we did using 60 and siRNA, we also measured the concentrations of short-chain fatty acids and the expression of these two biosynthetic enzymes to determine whether exercise influences the capacity for our microbiome to produce short-chain fatty acids. So what about exercise in the human gut microbiome? So here are some titles of some of the studies that have been done before we did our study. And you can see that some of these studies, for instance, differences in gut microbiota between women with an active lifestyle and sedentary women, and some between sedentary people and competitive cyclists. But I think all of these studies are cross-sectional, and that's a problem because we're comparing against different people, and we know that people who exercise regularly are very different than those who don't, and certainly they eat differently, and so that could be one of the causes for these community differences in the microbiota. So there really, at this time, before our study, was a need for a repeated measures longitudinal exercise trial where we carefully controlled diet, and we asked the question, could we independently affect the gut microbiota? And so I had two great students at that time, shown here, Lucy Mailing on the left and Jacob Allen on the right. So these two students spent about 2 years of their lives doing a study called Fit Gut, formulating exercise intervention trials for the gut. And the primary aim of this study was to understand whether endurance-based exercise training for 6 weeks could initiate a shift in gut microbial communities and some of the metabolites in previously sedentary lean and obese adults. So we had both lean and obese adults in this study. So here's just quickly some of the inclusion and exclusion criteria. You can see lean under BMI under 25, obese BMI above 25. They were all sedentary at the start, free of gastrointestinal disease, not having taken antibiotics or medications that messed with the gut or the gut microbiome. We did careful dietary control, as I'll outline in the next slide. So in this study, we had 18 lean and 14 obese, with 9 female in the lean and 11 female in the obese. They're all sedentary. We did some pre-testing, as you do before any intervention. We had an exercise period where they exercised 3 days a week at about 60 to 75% of heart rate reserve, so fairly moderate exercise for 6 weeks. And importantly, we wanted to make sure that whatever changes we saw with exercise disappeared when these people reverted to a sedentary lifestyle. So we asked them to, after the 6-week period, then stop exercising for 6 weeks. And we sampled fecal samples before the exercise, after the exercise, and then after the 6 weeks wash-up period. We did dietary analysis, careful dietary control for 72 hours before each fecal sample. We collected blood. We did body composition analysis. We did fitness testing. We did that before the exercise and after the 6 weeks of exercise, and then again after the wash-up period. So we controlled for diet by having them eat basically the exact same thing for 3 days before each fecal sample. And in that way, we did as best we could, maybe opposed to putting them in a metabolic ward and feeding them ourselves, control for diet so that we could say that any changes that we see really are due to the exercise and not differences in acute or chronic dietary intake. Interestingly, in the washout period of our 32 subjects, 7 people wanted to continue to exercise. So who are we to say they had to stop exercise? We would like them to, but so 7 of the 32 were non-compliant, which is good, I guess, but also reduced our power for that last part. So what do we measure and how do we do it? Well, we looked at metabolites, including our short-chain fatty acids. We looked at the functional genes, so we isolated the bacterial DNA and then searched for B coat and MMDA, and then we did 16S rRNA sequencing. We calculated delta values, both delta between pre and post exercise, and then also after the exercise and the washout period to be able to do things like principal components analysis, factor reduction, and then comparing, changes in the microbiome and the metabolome with the phenotypic changes that we saw in response to the exercise, like changes in body composition, changes in fitness, things like that, to see if there was any relationship between changes in the microbiome and changes in fitness that we could then explore in a more mechanistic fashion later on. So here's just some baseline characteristics of our subjects. You can see body fat was different, bone density was different, fitness was different. So here's some of the data. So we did show, so what you'll see here is E6 means post-exercise, W6 means after the 6 weeks of washout, lean is in the hatched and obese is in the thick black bar. And you can see that the 6 weeks of exercise intervention significantly reduced percentage of body fat, about 1% in both groups, increased lean mass in both groups. And you can see, importantly, that after the reversion to sedentary behavior, these adaptations were lost. Fitness changes as measured by maximal oxygen uptake were significantly elevated after the 6 weeks. We did not measure that after the washout. So I guess the conclusion from these data are that the intervention did as intended. Importantly, these body compositional changes in fat and lean mass occurred in the absence of differences in overall body weight. We did not want this to be a weight loss study, and we sold it as such to our subjects. This really was an exercise study. So in order to assess the microbiome, we need to use visual representation of the community structure. And one way to do that is through principal components analysis and using a PCOA plot. And so it's not simple, but the bottom line is that each dot in this graph represents an individual's bacterial community. Dots that are closer together are more similar. Dots that are farther apart are more different. And so here you can see the pre-exercise baseline data. So our lean are shown in red, our obese are shown in blue. And what I think you can clearly appreciate is that these people are different to start with. So there's a significant difference between the microbiomes of lean versus obese people. This is not a surprise. This had been shown before. I think we all appreciate that obesity is associated with an altered microbiota. But the interesting thing was, after exercise, looking at this same PCOA plot, we noticed that this difference no longer existed. In other words, there was a shift in microbial communities induced by exercise in both lean and obese, so that they looked a little more similar. When we looked at the washout period in terms of their microbiomes, again, there was no significant difference. And when we plot that as, we reduce the factors as an exercise factor or washout factor and run an independent t-test, we can clearly see that in the top panel, between the pre and the post-exercise period, there was clear orthogonal shifts in the microbiota between the lean group and the obese group. And then in the post-exercise period, between right after the exercise and then after the 6 weeks of washout, there was clear orthogonal shifts between the lean and obese group. So the conclusion here is that the lean differ from the obese at baseline, that exercise led to different types of shifts in the microbial community structures in lean versus obese, and that these changes occurred in the washout too, but they were orthogonal as well, so different. So what bacteria changed in response to the exercise in the lean and obese? So in this slide, you'll see that some of those bacteria, those in green are the ones that increased. Those in red are the ones that decreased. So you can see here For example, interestingly, some of these bacteria like Clostridiales and Lactospira are known butyrate producers, Rosburgia too, known butyrate producers. So that was fairly interesting. You'll also notice that, you know, the ones that changed in the lean group were different than the ones that changed in the obese group. And that's probably because they have differences to begin with, would be my guess. We also wanted to correlate the changes that occurred during exercise with the changes that occurred post-exercise. So what the reason why we did this is if for instance, say one bacteria increased during exercise, what we would expect is that after they went to a sedentary behavior, that same bacteria would decrease, and that's represented by a negative correlation. So if we correlated the different scores, we should see negative correlations, and that's indeed what we see here. You can see significant negative correlations for many of these bacteria taxa, indicating that whatever went up with exercise went down in the sedentary period afterwards, or conversely, if it went down during exercise, it went back up in the sedentary period after. So again, clear indications, strong data that indicate that exercise is having an independent effect, and when you stop exercising, that effect is lost. And so, you know, the bad news is that it doesn't hang around for a long period of time. It's gone at 6 weeks. We haven't done the studies to show actually how long it lasts. Is it 1 week, 2 weeks, 4 weeks? We haven't done that yet. I guess I'll skip that part. So the bottom line is that we see that exercise, 6 weeks of aerobic exercise at a very moderate level does change the community structure. And when you stop exercising for 6 weeks, that is reverted. We also looked at short chain fatty acids in these fecal samples. And you can see here graphs for acetate, propionate, and butyrate. I think the bottom line here is that you'll see that the lean folks, in response to the exercise training, realized bigger increases in short chain fatty acids than the obese folks. And that's a little bit, that was predicted a little bit based upon the microbiome analysis and those increases in the short chain fatty acids. I mean, excuse me, the short chain bacterial producers. Looking at the biosynthetic enzymes, B-coat and MMDA, you can see that there was some exercise effects here. There's an interesting main effect for obesity, demonstrating that at least the capacity to produce butyrate is elevated in obese individuals, but it looks like it is elevated in response to exercise as well. In the insets here, you can see that when they revert to sedentary behavior, a lot of these effects are lost. So for instance, propionate is reduced back down, butyrate is reduced back down. And same thing with the biosynthetic genes. So showing that something can change in one direction with exercise and then go back in the other direction without exercise is pretty powerful. So in conclusion, we showed that exercise increased and washout decreased short chain fatty acids, more so in the lean than the obese. That exercise increased the short chain fatty acid genes to generate them, and that obese people had higher B-coat. And then what we did is we related changes in butyrate, B-coat, and the bacteria that produce butyrate with exercise-induced increases in lean body mass. So when people exercise, they gain lean mass and lose fat mass. And so we related some of these changes in to each other, and we found significant correlations. So for example, changes in butyrate in the feces correlated to changes in lean mass to the tune of an R value of 0.875. Same thing with B-coat, same thing with several of the short chain fatty acid butyrate producers. So what we're thinking here is that perhaps when you exercise, you're changing your microbiota to increased energy harvest as a defense to the increase in energy expenditure that you realize during exercise, and also maybe to help change with body composition, which also costs energy. Lean mass expends more energy as well. We certainly need to follow up on that, but teleologically it makes sense that when you exercise and expend a lot of energy that you'd want your microbiota to change so that you can harvest more energy from the food you eat. That'd be great for a lean person, but maybe not so great for an obese person who's trying to lose weight, which is maybe one reason why exercise alone doesn't appreciably combat, obesity. You need diet and then exercise to maintain a proper body weight in the long run. So the take-home message here is that 6 weeks of moderate endurance exercise independently affects gut microbes, changes who's there, changes what they're doing. They produce more of the short chain fatty acids, and that there's some relationships between short chain fatty acids and gains in lean mass. Of course, these are just relationships. So it's interesting, but they're not cause and effect. And so we certainly need experiments to understand the mechanistic roles of exercise in terms of gut microbiome and metabolome. And so this paper got a lot of press when it came out, really the first study to show that exercise could affect the microbiome. Gretchen Reynolds, a health writer for the New York Times, asked a really insightful question. Is that one reason it's so good for us? Is exercise alters our microbiome? Is that one reason it's so good for us? And it's insightful, really, I think. And in other words, are exercise-induced changes in our gut microbiomes responsible for the health benefits of exercise? It's a difficult question to answer, no doubt. But I think the technology that we have to answer it was inspired by a movie in 1976 called The Boy in the Plastic Bubble. If you weren't around in 1976, maybe you're familiar with the Seinfeld episode where they visited the boy in the plastic bubble. So this is a situation where somebody's born with a defect in their immune system and they have to live in a sterile environment all their lives or they'll die because their immune system can't combat pathogens. So the tool we have to study this in a mechanistic way are called germ-free mice. So they're raised in a sterile environment. They've never been exposed to microbes. And so using this tool, we can introduce microbes into them to see the effects on their health and their development. And so it's a very powerful way to kind of test things mechanistically. And so in an animal study that we published in 2018, we did just this. And we addressed the question, does a transplant of an exercise biota affect colon health? And we did this in conjunction with Mayo Clinic because at the time, we actually didn't have a germ-free facility on our campus, but they did have one at Mayo and we had been collaborating with them. So we teamed up with them. So the study design, basically we had sedentary animals and animals that we exposed to a wheel for 6 weeks. After the 6 weeks, we harvested their biota. We transplanted that biota into germ-free recipients. Remember, these recipients did not exercise. So basically, we were trying to transplant the effects of exercise just by transplanting the biota. And really what we focused on was colon health, which makes sense. That's where most of the bacteria reside in your gut. And so that would be a good first place to look. And we looked at colons, normal colons, but also colons that we challenged or animals that we challenged with DSS. So in the absence or presence of DSS. And to summarize a lot of data, which I'll show you quickly in the slides after this, or at least breeze through them quickly. Amazingly, the animals that received the exercise microbiota had overall healthier colons. They had reduced colon inflammation. They had potential for improved gut barrier function. We found histologically that the animals that received the exercise biota had goblet cells that were full of mucus. They had reduced symptoms in response to the experimental colitis. They had attenuation in body weight loss, bleeding, and diarrhea. And in the colons of those animals that had received DSS, the colitis, there was an increase in signals of healing after the colitis, indicating that maybe the gut microbiota that we transplanted was helping them recover from the colitis quicker than the animals that had the sedentary biota. And I know I'm running short on time, and I want to certainly make sure I give Noah some time here. But this slide just shows you the differences in the gut microbiomes in the donor animals here on the left. So clear differences, like we've shown before, between animals that exercised and animals that were sedentary. And importantly, when we transplanted these biotas into germ-free mice, there were still differences in the microbiomes. As shown here. That's important to demonstrate. So the transplant worked. We did show that this transplant of the exercise biota reduced gut inflammation. So one thing that happens when you introduce microbes into an animal that's never seen them, as you might imagine, there's a profound immune response to the microbes, profound inflammatory response. And so this is like, several weeks after the transplant, and you can still see inflammation in some of these guts of the animals that had received the transplants, but the inflammation was lower in the animals that received the transplants from an exercised animal. Histologically, as I mentioned, some gut mucus depletion was lower in the exercised animals. Inflammatory cell infiltrate in the colons was lower in the exercised animals. I won't show this slide, but in the DSS model, we showed that colon length, which is a proxy for inflammation in the colon, was lower in the exercised animals. Body weight change was not as dramatic in the animals that received the exercise biota, as well as some other data there. And I'll skip this as well. So we concluded that exercise changed the gut microbiota, and that led to altered composition, short chain fatty acid profiles, and body weights in the recipient animals. I did show some of that. The short chain fatty acid profiles were strongly correlated with body weight and reduced gut inflammation in these animals. And exercise-induced changes in the gut microbiota attenuates DSS colitis outcomes and speeds regeneration while promoting antimicrobial defense. We actually measured some antimicrobial peptide differences. So kind of to summarize, we're very much focused on the role gut microbes play in the gut-brain axis and whether or not gut microbes contribute to the beneficial effects of exercise. We know exercise affects all tissues: skeletal muscle, adipose, liver, the immune system. We've shown you some data today on the gut, and also the brain. The brain is important. I haven't shown you a lot of data on that, but that's kind of where we're going, and Noah's going to speak to that in a minute. So I want to say that you are what you eat, certainly, but you also are what you do. And so it's important to look at all types of lifestyle behaviors: diet, exercise, and it does appear that both of those can impact your gut microbes and lead to changes in your health. So with that, I'd like to acknowledge students who've been in my lab: Jacob, Lucy, Yi, Noah, who's currently there, the army of undergraduates that help us, the tech folks and support folks at our campus, people at Mayo Clinic who helped us with the exercise germ-free studies, as well as my colleagues at the U of I, many of them in the Division of Nutritional Sciences: Rod Johnson, Rex Gaskin, Sharon Donovan, whom you'll hear from later in this seminar series, as well as others. So thank you for your attention. I suppose we have time for maybe one question or two, but we certainly can take questions after the after Noah's section. So I'll leave it up to Marie.
Okay, thank you, Dr. Woods. I think in the interest of time, we'll go directly to Noah. So I just wanted to introduce our final speaker, Noah Hutchinson. He is a second-year USDA fellow in the Division of Nutritional Sciences, and currently Noah works, as you saw, in the lab of Dr. Jeff Woods, and his work investigates how diet and exercise modulate the gut microbiota and ultimately link to host health. So we will turn the control over to you, Noah.
All right, thank you for that introduction. I would also like to thank everybody for coming out today, and I want to thank ILSI for putting on this event and DNS for allowing me to have the opportunity to give this talk today. So as Dr. Woods said, I'm going to be giving kind of an expansion on what we have going on at the moment, and then talking about some future directions where we may take it. So firstly, what we have going on here is a logical extension to the Fit Gut study, where we are performing an exercise intervention for ulcerative colitis, Crohn's, and IBS patients. This is a collaborative project with Carle Hospital, and thanks to COVID, we are currently still in the recruitment phase for it. That's been a little bit of an obstacle to jump over. What we're looking for are adults who have a confirmed diagnosis of an inflammatory bowel disease that are currently sedentary, not pregnant, and obviously haven't taken antibiotics within the last couple months. So before our testing period, we're going to run some screening on them. Obviously, we need to get their medical history and do some baseline measurements of fitness, like VO2 max and a DEXA for body composition. We're going to review their diet history and then work with the dietitian to create a 3-day menu that will allow us to control microbiome to the degree that we would like to. We'll also be taking baseline blood and fecal samples to assess certain biomarkers of interest. So with this study, you see it's a crossover design. So that way we can determine if the adaptations will revert after cessation of the exercise period. So we're going to be doing repeated measures of fecal samples, certain questionnaires, blood samples, lactulose, mannitol, gut permeability tests, and we'll be closely monitoring their diets to make sure that everything is consistent. The exercise protocol is graded in that it increases in time and intensity as it goes on. This will help us kind of, increase lean body mass, but not decrease overall body mass, kind of like you saw in the Fit Gut study. That's kind of the goal of this type of protocol. So the question now is, how does this relate to the brain, right? This seminar series is about the brain. It's been quite well documented for quite some time now that patients with inflammatory bowel disease exhibit increased rates of many psychiatric disorders. Specifically, depression is very prevalent among them. They generally exhibit high levels of pro-inflammatory cytokines, which act on the gut-brain axis and microglial cells. They induce the rate-limiting enzyme of the kynurenine pathway IDO1 and create potential neurotoxic kynurenine catabolites, which potentially also reduce systemic tryptophan availability for serotonin biosynthesis, which may exacerbate sickness behavior. And I'll talk about this a little bit more later. But back to the microglia, the inflammation certainly does have an effect on pro-inflammatory activity in microglia. So we know that I'm expanding on the study that Dr. Johnson brought up. We're working with them. Dr. Johnson is the PI of this grant. We know that aged microglia are pro-inflammatory and very hyposensitive. We also know that aging increases intestinal inflammation and viscerosensory signaling to the brain. We also know that gut microbial production of short-chain fatty acids downregulates this pro-inflammatory microglial activity. So in this grant, we're using fiber feeding strategies to modulate the intestinal inflammation and ideally microglial inflammation. And we're going to be measuring local ileal and intestinal inflammation as well, and markers of neuroinflammation and neurodegeneration using histology, fluidigm RT-PCR, metabolomics, and other advanced techniques. Another interesting aspect of the study is the group is working on developing a transgenic free fatty acid receptor 2 knockout model. This will allow the group to test its mechanistic properties in this observed phenomenon. Understanding these mechanisms will be very helpful in developing dietary interventions to combat neurodegenerative states, activities, and conditions in the future. The first aim is to determine, do intestinal short-chain fatty acids normalize communications in this viscerosensory pathway and inhibit age-induced pro-inflammatory microglia? And secondly, does this happen through free fatty acid receptor 2 on intestinal epithelial cells? So moving on, we know, based on what you heard just now from Dr. Woods, we know that microbial composition can be altered by consistent exercise training. And we know that fecal microbial transplants can improve gut physiology and reduce symptoms in response to colitis when compared to microbiomes from sedentary animals. And there's some human versions of this in the works right now. So now the question is that we thought was very interesting would be to investigate would be, do gut microbes contribute to host adaptations to exercise? The University of Illinois recently installed a rodent notobiotic facility. You can see a couple of the couple of machines here. So we thought this would be a good way to capitalize on this installment. So the first experiment to measure this question, to test this question is using a notobiotic model. So the first aim is, do germ-free mice display the same voluntary wheel running patterns as conventional mice? And the second aim is, if they do end up running the same amounts, do germ-free mice adapt to exercise similarly or equivalently when compared to conventional mice? So to test this, we're using a 2x2 model here that will help us tease out the differences. Tease out when certain effects are related to exercise or certain effects that are related to the notobiotic mice. So for 8 weeks, some of them will be exposed to either sedentary conditions where they'll just have a locked wheel in their cage, or some of them will have a wheel in their cage that they will run on, as mice just do instinctively. And we have a telemetered system that allows us to monitor when and how much they run. We'll be monitoring their body weight and food intake On the 7th week of the intervention, we will be measuring their glucose tolerance via an IP injection test, which I'll talk about a little bit later. And at the end, we're going to go doing some physical fitness measures like grip strength and treadmill endurance. And then upon dissection, we will use tissue collection for muscle enzymes, colons for gene expression, brains, and many other tissues. We'll also be measuring their body compositions at the end. Germ-free research has quite a few limitations to it. As you can see in the background here, that's one of the isolators. There's not a lot of space in there, and your manipulative capabilities are severely hindered. Germ-free mice also experience altered development, so it's not a completely accurate depiction of what will actually happen. Not only is it an animal model, but it's also an abnormal animal model. These are more expensive mice to work with, that's pretty obvious, and it's more time consuming. There's a lot of stipulations with entering the germ-free facility and getting equipment into the germ-free facility, which doesn't allow you nearly as high level of manipulation either. If you look at this hood, I'm just bringing this back to kind of show you that this is really all the space you have to work with. So manipulations have to fit within there. And so I don't think our treadmill is going to fit in there. So repeated measures are difficult. However, that does not obviously invalidate results. So this is just a kind of a evaluation of what might happen in aim 1. So it's possible that the mice run less. And this would explain obviously the lack of adaptations in the second aim, but will be interesting to follow up on. Potentially introducing a microbe to these mice could make them run more. That would be an interesting phenomenon to investigate. And if they run more, well, that's going to be a whole slew of other interpretations, and will be interesting to follow up on. The probiotic approach is not something unique to us. There have been other studies that have used this approach, attempting to enhance performance with probiotics. However, they're still in early stages and efficacy is still somewhat unclear. So the second model of doing this is antibiotic-induced dysbiosis, where you administer broad-spectrum antibiotics to the mice regularly. So the aims are very similar to this in this experiment. The first aim would be, does regular consumption of broad-spectrum antibiotics affect the natural wheel running behavior? And the second aim would be, does this consumption of antibiotics affect the adaptations that they will experience to regular exercise? Study design is very similar. This is the study design we just used for a recent cohort. It's important to note that we added the antibiotics 3 weeks in because that allows us to determine whether or not the mice, their running wheel patterns are affected by the antibiotic administration. There's a decent amount of variability within the data as a whole, so it's important to get a general pattern before you add antibiotics to see if the antibiotics had any sort of effect. So we're going to be collecting fecal samples, taking fitness measures, doing a glucose tolerance test, and testing their treadmill endurance at the end as well. We'll also be collecting all sorts of other measures for tissue, biochemistry, and omics approaches. So the feces will be used for 16S sequencing for the microbiomes, as well as a metabolomic analysis for short-chain fatty acids and potentially other microbial metabolites. Body composition will be done before, after, and after the lead-in and at the end of the study. However, this is not currently available. So what I'm going to talk about today, we actually had to use perigonadal fat pads as a replacement, but that is also a marker of fitness and voluntary wheel running adaptations. And like I had previously stated, we'll be doing muscle enzymes and gene expression, as well as protein expression, histology and colonic gene expression, glucose tolerance testing, brain gene expression. Heart weight adaptations are generally observed in exercise models, as well as serum analysis. This is the general procedure to conduct a glucose tolerance test in mice. After a 12-hour fast, you administer an IP injection based on their weight with a glucose solution, and then measure their glucose slightly before the injection, and then 4 times 30 minutes, each 30 minutes apart after that. And you kind of measure the time under the curve and the disappearance rates of the glucose. What's interesting about this test with concerns to the antibiotic treatment is that there have actually been models in the past that have tested this in an antibiotic depleted model, and they've seen increases in glucose tolerance without exercise. Generally, exercise interventions for glucose tolerance are done on mice in a high fat or some other obesogenic conditions. But interestingly, the antibiotics actually increase glucose tolerance in these mice. And the authors of the paper theorized that potentially that in the absence of short chain fatty acids, as Dr. Woods mentioned, short chain fatty acids are used as fuel for colonocytes, but in their absence, colonocytes may shift towards glucose utilization instead. So for AIM-1, being the effects on voluntary wheel running, our hypothesis was no. The reason we hypothesized this is because there's a recently uncovered relationship between ghrelin levels and wheel running activity. So ghrelin levels were actually shown to spike right about when the dark cycle kicks on for mice, which is actually when they start to become the most active. Ghrelin, and they also developed a ghrelin knockout model, and ghrelin knockout mice actually ran less and overall and during the dark period than wild type. And single administration of a ghrelin receptor agonist in the ghrelin knockout mice at the beginning of the dark cycle actually markedly enhanced their running. Another interesting note here is that time-restricted feeding actually increased wheel running in both groups. And ghrelin knockout mice showed attenuated postprandial enhancement of dopamine. Potentially, there's a relationship here between the reward circuit, but more research needs to be done to prove this. Now, why is that interesting? So, that connects to these actually broad-spectrum antibiotics model I just referenced previously with the insulin. They actually measured ghrelin levels and did not see a difference in ghrelin production in these mice. This leads us to believe there will not be a difference in ours. So to test this, this is a picture of the telemetered running wheel in the cage. It sends signals to that hub that's in my hand right there, and the computer will it'll show up in a computer program, and then we can export it for analysis. Here's some preliminary data from this first cohort we actually did. So here we see that we didn't really see an effect based on antibiotics. So that's a pretty easy interpretation there. You'll see it's pretty variable in general. That's just kind of the nature of the data, but we did not see an antibiotic effect. So the next aim, testing whether or not the adaptations will be differently. We hypothesized that they will have different adaptations. And then this is a picture of our treadmill for our endurance testing. So in our first cohort of mice, we did notice a decrease in body weight due to voluntary wheel running. So there is some sort of adaptation happening there. Interestingly, we did also notice a decrease in perigonadal fat pad weight. So it's not just a reduction in body weight, it's also a reduction in fat pad weight. This indicates that their body composition might be trending favorably as well. So here is our treadmill data. This is our test to fatigue. We have the y-axis is the amount of distance run. We did a graded protocol. You'll see here that there's quite a big training effect in the voluntary wheel running mice. Their endurance capacity is quite a bit higher. We did not see an antibiotic effect in this measure. However, it's important to note that we only administered the antibiotics for 3 weeks rather than the whole 6 or 7 week trial period. Perhaps a longer administration of antibiotics during the wheel running would potentially show larger effects, especially because these mice had 3 weeks to run and adapt before they were given antibiotics. We also, potentially a larger cohort would yield more significant results. So our early interpretations of these results are that physical fitness tests show that we had a successful running wheel intervention, that putting them on the wheels is doing something to their physical fitness. Up next, we have to do microbial sequencing, metabolomics, and muscle omics to reveal a more in-depth look at what's actually going on underneath the skin. And future cohorts will include longer antibiotic treatment periods now that we know it doesn't affect voluntary wheel running. And obviously, we're going to try to get a larger N in the future. So the question related to all this is, what about the brain? Where do we go from here? Obviously, this is the gut-brain axis we're here to talk about, and there are some interesting directions we can go with this. But it's pretty well documented, and Dr. Woods touched on this already, that exercise has been proven to slow progression of neurodegenerative diseases. It promotes neuroplasticity and neurogenesis via brain-derived neurotrophin factor. It's also been shown to improve aspects of cognition and to treat neurological diseases like depression and anxiety. Now, this is particularly interesting because, as I said previously, these are very commonly observed in patients with inflammatory bowel diseases. However, the mechanisms that are proposed, most of them lack a whole lot of support and evidence. So this is an interesting route of investigation that needs to be explored more. So at the forefront of this, there are some gut microbial metabolites that have some interesting effects. I had mentioned this previously. So there are associations that have been drawn between the composition of the gut microbiota and symptoms of depression. IBD patients, which I previously said are frequently depressed, display increased IDO1 activation and decreased levels of fecal tryptophan-derived aryl hydrocarbon receptor ligands. And aryl hydrocarbon receptor ligands have shown efficacy in ameliorating depressive symptoms in some models. This is a relatively new area of research, so there's not a whole lot of research on it, models on it yet, but hopefully there will be more coming soon. This is a model from Dr. Wood's presentation. The exercised biota transplants did decrease inflammation and downregulated IDO1 pretty significantly. So a proposed mechanism I have for you here is that, so it's been well documented in exercise that exercise upregulates muscular expression of PGC1 alpha, the mitochondrial biogenesis gene. And this mitochondrial biogenesis gene actually upregulates kynurenine aminotransferases. So IDO1 is the rate-limiting enzyme in the kynurenine pathway of tryptophan catabolism. Kynurenine is a precursor for multiple potential neurotoxic compounds such as quinolinic acid, among others. So decreasing the amounts of these kynurenine compounds in the blood may potentially promote brain health and ameliorate depressive symptoms. Exercise may potentially promote microbial production of AHR ligands, as well as decreasing inflammation, as we've seen previously, which will also downregulate IDO1, as that is induced by pro-inflammatory cytokines. So in the serum, you have, so due to this reduced IDO1 activity in the serum, you would hope to see increased availability of tryptophan. Tryptophan is important because it serves as a biogenic precursor for the neurotransmitter serotonin, which we know is associated with good affect and positive mood. So with the exercise, we would hopefully see a decrease in serum kynurenine. With the upregulation of this kynurenine aminotransferase that can convert kynurenine to kynurenic acid, you would hopefully see an increase in kynurenic acid, which actually exerts neuroprotective effects. So with this increase in serum tryptophan and decrease in kynurenine and increase in kynurenic acid, you would hopefully see in the brain increased levels of serotonin and melatonin, which is also a downstream of tryptophan, and decreased levels of these neurotoxic compounds that I've said before. Now, what would happen if we did antibiotics treatment or if we did a germ-free model here? So you obviously don't have the microbial production of these aryl hydrocarbon receptor ligands. So what would happen to inflammation is hard to tell. IDO1 activation obviously would be influenced by that removal. I would say that the muscular adaptations would still somewhat exist. However, the magnitudes I'm unsure of. And well, I'm hypothesizing here. And the tryptophan and kynurenine ratios may be affected by the presence of the gut microbes. And the outcomes would potentially be different, but this remains to be seen. It is an interesting area of investigation moving forward. So the question is, where does nutrition come in? So there's obviously the potential for consumption of microbiota-enriched prebiotic fibers to synergize with exercise. It'll be important to tease out whether these effects are additive or synergistic with some 2x2 designs. As we've seen that exercise enriches the microbiota, and we've seen that dietary fiber enriches the microbiota. Would these effects stack on top of each other, or would they magnify each other and intensify these results? There's also some similar studies in probiotics that could serve as similar functions to diet and exercise. And fecal microbiota transplant models for depression are still relatively early, but would also be interesting to look at. And with that, I am done, and we'll leave it up to the floor for any questions. Thank you for listening.
Great, thank you so much, Noah. And we do have a few minutes for questions. So I'm going to start with kind of a big picture question. And Noah, you just talked about the intersection of diet and exercise. So just thinking more broadly, what do we know at this point about the contribution of diet versus exercise? Could you choose one? Do we know the degree to which this might be additive or synergistic?
You want to take that one, Noah, or you want me to give it a stab?
I think you would be better suited to answer that question.
So that's a great question, and this is aspirational where we want to test this. So I think the existing data on the effects of diet on the microbiome versus the effects of exercise suggests that the effects of diet can be much stronger than the effects of exercise. I think that can be said, at least on a community structure level. To this point in time, however, there's very little study of the combined effects of diet and exercise, so we don't know if there's any additive or synergistic effects. I believe there's going to be, because I think that the diet and exercise affect the microbiota differently. If you think about diet, you're thinking about substrates that come in to the lumen of the gut that interact with bacteria and niches of bacteria within the gut. I think with exercise, and again, this is the studies that we've done have controlled for diet, there's probably some either something leaking out of the body into the lumen, perhaps lactate, or there's some neural mechanism that's activated with exercise that communicates through the enteric nervous system or through the immune system to alter the biota. And so, it's my belief, I guess, I don't have data to back this up, that because exercise and diet likely interact with the biota through different mechanisms, that there could be additive or synergistic effects when applied together. There just hasn't been real good studies that have done, aerobic exercise with and without fiber, and or other dietary interventions to look at that. So good question. Yeah, my hope and guess is that there's going to be additive and synergistic effects, and doing both is better than doing one. I think certainly for human health, we know that.
Right. Okay. Thank you, Dr. Woods. So talking about the physical activity side, a question about how you decide, either for animal or human studies, on the physical activity intervention. So do you choose that based on existing evidence that this specific, 3 days a week or this specific heart rate has an effect? Or how do you choose that? What do we know about that physical activity that might have a positive effect at this time?
Sure, these are questions we get all the time. So it's really a very practical question, an important question. I mean, what do I need to do in order to affect my health or my gut microbiome? And so the studies that we have done, our strategy pretty much, with people has been to focus on exercise that is moderate in intensity, that starts at low levels and increases, especially if we're taking sedentary subjects or people into our exercise studies. So in the Fit Gut study, for example, this was a very moderate exercise that built up over the course of 6 weeks because they were sedentary. It was cardiovascularly based exercise, so it was aerobic exercise. I think maybe we needed to point that out a little better in our presentation. So that, you know, we're talking about aerobic exercise here. We're not talking about resistance exercise or strength training. There's very few studies that have looked at strength training and the microbiota. And I don't know if we can come to any conclusions yet, but I bet you there's going to be some effects. But certainly there are, and we've shown it with endurance-based training. So, you know, going out 3 times a week and doing, you know, half an hour, 45 minutes is what we have shown to affect the gut microbes. You know, would longer be more, you know, better, or, you know, we don't know yet. There is some evidence that high intensity interval training or HIIT training also can affect the microbiome.
Okay, that's interesting because that is a little bit of a combination of both cardio and strength. Okay, so another question about the exercise-induced changes. How much do you think is related to changes in GI transit time versus, you know, we know that exercise affects host metabolism?
Yeah, so another great question there. There is some evidence to suggest that regular exercise and physical activity can speed up fecal transit time. So the time it takes for, you know, food materials to move through the gut. But that's not universal. There's also some studies that haven't shown that. And so, you know, I think, you know, maybe is the answer. Maybe there's a speeding transit time. Some believe that faster fecal transit times are responsible for lower colon cancer risk in people who exercise regularly, which makes sense. I mean, if your feces are in contact with your colon wall for long periods of time, some of those carcinogens can hang around and cause transformation of cells. And that's certainly been touted as a potential mechanism, but I don't think it's that simple. I think there's many things that are going on when we exercise, and in particular in our gut, that can help protect us from colon cancer and maybe from IBD, although we need to do those studies.
Okay, super, thank you. With attention to time, maybe we'll just take one more question. Well, this is kind of a follow-up, but what do you think about the changes in the microbiota being due to alterations in the blood supply, which happen during exercise?
Yeah, sure. Another good question, and you know, everybody's thinking along the same lines as we are. What does the exercise do to the gut that we know of that could explain some of these changes in the microbiome? So when you exercise, there's a shunting of blood away from the gut to the muscles and skin, and so there can be, in some instances, hypoxia of the gut. Usually it's not severe and not a problem in moderate types of exercises, but it can be a problem if the exercise is intense, prolonged, and especially if the exercise is in the heat, because that puts a greater strain on your cardiovascular system where you shunt even more blood to your skin and away from your gut. And so you can get frank hypoxia in the gut. And some people do present clinical symptoms, say after a marathon, where they'll have you know, symptoms of diarrhea and problems in the gut because of this hypoxia. So it's like a transient colitis, I guess, due to hypoxia. In terms of that being responsible for changes in the gut microbiota, I don't know. I mean, most bacteria in the colon are obligate anaerobes, so a lack of oxygen should not affect them too much. In fact, it may help them. There is an emerging literature that higher levels of oxygen in the lumens of our colon, perhaps due to gut barrier disruption, may actually lead to an outbreak of facultative anaerobes, bacteria that can switch their metabolism from anaerobic to aerobic. And many of those switches or many of those facultative anaerobes are associated with taxa that are opportunistic pathogens. So that could be a bad thing if it went on too long. But you know, as for the mechanism, you know, it's a good idea, but it's very difficult to test in terms of ruling that as one of the sole factors whereby exercise affects the gut microbiota.
Thank you. Okay, fascinating stuff. I think we're just about out of time, so we wanted to thank everyone who's stuck with us. Thanks to the University of Illinois and ASN as well. And I just wanted to, thank Dr. Johnson, Dr. Wood, and Noah. And Noah, we look forward to seeing the outcomes of your work. And I just wanted to point out that we do have a webinar for the next 4 weeks, so please visit our website to register. The next one in our series is shown here, and that will be with Dr. Kelly Swanson and Celeste Alexander, and it's at the same time next Thursday due to the holiday weekend. So thanks everyone for joining, and we hope to see you in our next webinars. Have a good afternoon.
Thank you, everyone. Bye-bye.